Yes, good morning to all. I am Dr. Jayakumari, Professor, Department of Pharmacognosy. Today I am going to take the topic of plant fibers. It comes under the Unit 5. The program is Pharmacognosy and Phytopharmaceuticals. Program formed the second year. The course name Pharmacognosy and Phytopharmaceuticals. Model of the title plant fibers. The objectives of the present slide: introduction to pharmaceutically important plant fiber, detailed description about the source, chemical constituents and the chemical properties, preparation of the fibers, and examples of the fibers: plant fibers, cotton, jute, hemp. Fibers are natural or synthetic substance. They are elongated with a pointed end, long strand-like structure. The fibers obtained from various sources, which can be categorized as based upon the source: plant fiber, animal fiber, mineral fiber, synthetic fiber, and regenerated or semi-synthetic cellulose fiber. Plant fiber example: cotton, jute, hemp and flax. Animal wool and silk. Mineral fiber: asbestos, glass wool. Synthetic fibers, they are nylon and terline. Sometimes the fibers are obtained from natural source. From modification, they are called viscose and acetate rayon. Cotton. It is synonym: absorbent cotton, cotton wool, surgical cotton. Biological source: it comprises of epidermal trichomes from the seeds of the different species of Osepium species, Osepium herbaceum, Osepium barbadensis, Osepium glyptotum, all belongs to the family Malvaceae. Cotton is a very easily grown in temperate region like India and Egypt, and the medicinally used cotton is called absorbent cotton. It consists of the trichomes, but these are free from the fatty cuticle. That is, cuticle contain fatty substance. In absorbent cotton, the fatty substance was completely removed by alkali treatment. Thereby, it can able to absorb the any impurities, bleached and sterilized material. After that, it was bleached and sterilized. Preparation. The plant after flowering period. The fruits are fully grown, and the fruits are a capsular type of fruits. They are collected from the plant when it is size about 2 to 10 centimeter in diameter, with brownish seeds. The white hairs are attached along with that one. The seeds are covered with a number of numerous holes. Along with the capsule wall, the fruits is called bolls. The bolls are collected during the autumn season or after the flowering period. Then they are subjected to the Gind process, a machinery process which is used to remove the separate the seeds from the mass of the fibers. The fibers, cotton fibers, are separated by using ginning process. After that, the fibers can be graded as per the size: short fiber, long hair, each other. Short hairs and are used for the manufacture of absorbent cotton, whereas long hairs are used for the preparation of the cloth. The raw cotton obtained from that procedure containing impurities, because it contains some fatty cuticle, waxy cuticle. Wax it also present, fat also present, coloring matter, and some other fragment of vegetable debris. They are somewhat dull white in color. It does not absorb any liquid, so they are called non-absorbent cotton. So after that, it may converted into absorbent cotton. The fatty material is removed by treating with dilute caustic soda for about 10 to 15 hours to remove the wax and other fatty substance, coloring matter. Then after that, it was bleached by using suitable bleaching agent. Then chlorinated soda is commonly used, or methyl hypochlorite can be used. After bleaching, it was repeatedly washed with water or weak acid for the neutralization of the excess of acid, and then again it is repeatedly washed with water to neutralize the acid. Then it was dried, then it can be graded into flat sheets known as surgical cotton, or otherwise it is known as absorbent cotton. Finally, the absorbent cotton was sterilized suitably and packed in a wrapper and sterilized with some radiation method. Here the gamma radiation method was used. This is the photography which shows the first one, capsular fruits. It is a bolts type of capsular fruits. Each fruit contains numerous seeds. When it is fully grown, tend to break. They are collected from the plant. Then they are broken into small pieces. From that one, the seeds are removed by ginning process. The third diagram, which is represents the ginning machine normally used. After separation, it was treated, then it was bleached, then it was sterilized, then come to the market in the name of absorbent cotton or surgical cotton. The absorbent cotton is most commonly used in surgical industry. Description: it is a white fragmented soft hair-like structure which is appear as a microscopic halo, unicellular, flattened trichome with a pointed end. It is about 25 to 35 micron in diameter, 2.5 to 4.5 centimeter in length. There is no odor, no taste. Raw cotton usually contain 90% cellulose, the remaining 10% may be fat, moisture, wax and protoplasm, remaining part of the protoplasm. Absorbent contains about entirely 99% of cellulose, only it contains little amount of moisture, about 7%. Identification test when treated with the N by 50 iodine solution and 80% sulfuric acid, absorbent cotton gives a blue color stain. Cotton soluble in 55% of sulfuric acid and insoluble dilute sodium hydroxide hydrochloric acid. With the iodinated zinc chloride solution, it becomes a violet color. This is a test which is used to differentiate the raw cotton from absorbent cotton. The test name is called coaxum test. Chemically, coaxum is nothing but copper oxide solution. Absorbent cotton uniformly swells and dissolves, whereas raw cotton swells unevenly with uneven swelling, dissolving indicate the balloon-like swelling. One more test is used to determine the raw cotton and absorbent is called absorbency test. For that, 1 gram of cotton was taken, which put in a volume of water, 20 ml volume of water, placed in separate 2 beakers. When it is absorbent cotton, it will absorb the water and sink at the bottom of the container. Whereas if it is a raw cotton, it does not absorb the water because of the fatty cuticle. It float on the water surface, which can be used by simple test to differentiate the raw cotton from absorbent cotton. Storage cotton can be stored in the double wrapped cover to prevent the microbial content. It's stored in a cool place. The improper storage indicates the brittle in nature cotton, friable in nature, and color was somewhat more color will be there. Uses, most of the cottons are used in surgical industry, absorbent cotton, whereas raw cottons are used in the preparation of cloth and other things, filtering medium and insulating material. Next is jute. It comprises phloem fibers, a stem of various species of Carcorus species. Carcorus oletorius, Carcorus capsularis family, Tiliaceae family. It is a yellowish brown in color, coarse in texture, about 1 to 3 meter in length. They are hygroscopic nature, good tensile strength. Preparation when the plants are flowering period, the straight stems are cut down from the plant. They are made into bundle. They are dipped in water. Then after that period, fermentation is over. They are taken out. Retting or bacterial disintegration takes place during choking of the plant. The bark gets loosened from the stem of the plant and the phloem fibers are get loosened from the bark. Then after that they are separated. Workers beat the end of the stem with a hard mallet or any instrument. The jute fibers are get separated from the bark material. The fibers are cleaned with water repeatedly. Then they are hang up in the robe in the sun to dry and it can be bleached. The jute fibers are then graded according to the color, fiber length and strength. This is a photography which represents the first one photography represents the jute plant. They are about 1 to 1 meter height. They are collected, they are made into small bundles along with the stem. Then from that one they are choked in water. This is called a retting process. They are choked in water, repeatedly turned up and down. During choking for one month or 2 months, they get separated. The bark fibers are loosened. The bark fiber then hang up in the rope in the sun to dry and bleach. Chemical constituent contains 50 to 53% of cellulose, hemicellulose 20%, lignin 10 to 11%, fat, wax and moisture. Identification test when treated with the fluoroglycinol hydrochloric acid, it gives red color due to the presence of lignified tissue, that is nothing but lignocellulose present in the cell wall of the fiber. Uses, it is used as a preparation of filtering medium, staining medium. It also used for the commercial industry for making of gunny bags, ornamental item. Hemp, it is Indian hemp, Indian cannabis maricoma. It is obtained from pericyclic fiber of the plant Cannabis sativa, Cannabis indica, family Moraceae family. It also collected from the plant fiber. It is somewhat a pericyclic fiber. So it collects from the stem of the plant. The stem was placed in water. Then it can be steeled with the help of teeth-like structure. Then you remove the capsules. The process known as rippling process. The stems are dyed in a bundle. They are immersed in water for about 14 days. And the process is known as retting. So after retting process over, during retting the fermentation takes place, which makes the tissues very soft and becomes a loose. After completion of the retting process, the stalks are dried, broken in a special type of mill, after which the xylem fibers are removed. The last trace amount of wood and other parenchyma, adjoining parenchyma can be removed by to make the fiber parallel. The hemp fibers are longer, stronger, more elastic and more absorbent and more resistant to the when compared to the cotton. Chemical constituent contain 57 to 77% of cellulose, hemicellulose 9% lignin. It also used for the preparation of robe, sacking and carpet and other knitting and webbing also. In surgeon it is used for the some insulating materials, some wire gauze and other things. So this is a cannabis sativa plant. Rippling using steel teeth comb. After rippling it should be taken into the rippling process, choked in water. After that the fibers can be removed, then it was dried and come for the market. Next, fiber is natural or synthetic, is elongated with a pointed end. Cotton is epidermal trichome from the seeds of different species of Gossypium containing cellulose usually, and jute comprises of phloem fibers. Fiber is a natural synthetic substance. It is a summary of the slide. Fiber is a natural synthetic substance elongated with a pointed end. Cotton is epidermal trichome from the seeds of different species of Gossypium containing cellulose as a filtering medium, surgical dressing, used insulating material. Jute comprises the phloem fiber of the stem of the various species of Carcorus. It containing cellulose, hemicellulose, lignin. It is mostly used for the medicated toes and filtering medium and staining medium. Hemp, it is a pericyclic fiber obtained from cannabis and other species of cannabis. It is also contains cellulose 77% and 14% hemicellulose, lignin 5 to 10%. It is used for making of robe, sacking, carpet and other netting and webbing structure. Thank you students. எவ்வளோ கஷ்டம் படுறா ரெக்கார்ட் பண்ணுறது.